Swayam Prabha. Digital India. Educated India.
Okay, so let us go from the first one. So here the main issue is that this fully reduced quinol, this QH2, like where my cursor is. So from this, one electron is transferred to cytochrome C, and the other one goes back via the cytochrome B to a fully oxidized quinone, and that becomes semi-quinone radical. Okay, so having given up both electrons and both protons, this becomes fully oxidized quinone. Then in the second cycle, one more molecule of fully reduced ubiquinol binds to this, and from there, through this iron sulfur protein, one of the electrons go to the cytochrome C. Okay, just like the first one. And the other one via cytochrome B, these two heme, returns to reducing this semi-quinone produced in the first cycle back to fully reduced ubiquinol. Okay. So while the electron has come back, but the proton has not, it is pumped out. And as a result, we take protons from the matrix side to fully reduce it to QH2. So that is the reason protons are taken from here. Okay. So here both protons are gone, and here again both protons are gone. And here it is not shown because this is radical, and that fully to reduce, you need to take the two protons. Went through these electron transfer carriers, and then we saw how the electron finally goes to reduce oxygen to water. So the electron transport part we have seen, and then we saw in the process the protons are being pumped across the membrane, as shown in this slide, to the intermembrane space. So today what we are going to do is we are going to focus on how this proton gradient, so the gradient being it's a high concentration in the intermembrane space and low in the matrix of the mitochondria. So once more, to get clarity on this, I'm going to draw the same mitochondrial cartoon, just for some of you who may not be so diligently following this. So if this is the outer mitochondria, so you have the inner mitochondria. So I'm not going to draw all the cristae. So this space, this is the intermitochondrial space. So this is where you have positive charge, and this is where you have negative charge. So this post, in the slide cartoon, the left side where the arrow points corresponds to this space, and where the arrow starts from, the right side is this inner side. Okay. Okay. So now the end result is we have a chemical potential energy here, because the chemical space is this proton. It has a concentration gradient. Second, you have a charge separation as well. Therefore, you have electrical potential energy. So these two components are the energy part generated by the electron transport downhill, from a molecule with the lower reduction potential to a molecule with the higher reduction potential. That downhill flow of electron resulted in generating these two components of potential energies. Okay. So now when the proton returns back to the matrix, you are going to have a free energy change, and that will equal this. Okay. So concentration on one side versus concentration on the other side, plus you have to take the differential in the membrane potential. Okay. So the Z is the actual electrical charge. In the case of proton, for one proton, it is just 1. So now if you take, instead of natural log, if you take log to the base 10, then it would really be pH. Log to the base 10 of C2 will be pH, right? The pH is the negative log of hydrogen ion concentration. So therefore, this equation will rearrange into 2.3 times RT times delta pH plus Faraday's number times the membrane potential difference. So this is the way we will obtain the free energy change in the downward flow of the protons. Okay, so this again brings us back to the chemiosmosis. So this cartoon is much more clearly drawn here. So this is the outer mitochondrial membrane. This is the inner mitochondrial membrane, where all these electron transport complexes that we learned are all present here. Okay. And so this is that cytochrome C that diffuses from this to this, and ubiquinon is within the membrane. So now as the electron flew from this to oxygen and becoming water, we have these protons pumped across by these 3 complexes. And so the numbers correspond to a single molecule of NADH, as I told you yesterday. And when you talk about a molecular oxygen, which is what is normally consumed, producing 2H2O, then you will start with 2 NADH and the numbers will double here. And now and when these protons come down, they actually come through this pump. Okay, there is no other way the protons can leak across the membrane. And this pump, this is more like the right analogy is like a hydroelectric power project. So you have a dam where the water has been stored, prevented from free flow down the river. So now you have generated potential energy. Now when you allow that water only through a narrow pipe, the pressure is going to be very high, and that high pressure, the potential energy difference from upstream of the dam to the downstream is now used to turn the turbine. Okay, because that's the only way you are allowing the water to come out. And the turbine, when it is in a magnetic field, it generates electricity. Exactly the same thing that happens here to the last detail of the turbine. Here again, you are going to have a rotation movement generated to produce the ATP molecules that we will see towards the halfway of the class today. So since this involves a chemical potential like pH difference, and then you have a transport process, okay, so to coming down gradient, and that is why Peter Mitchell chose this word chemiosmosis to signify that the process involves a chemical reaction as well as a transport process. Okay, so it is transported through this pump. And to account for both of this, he coined this word chemiosmosis. So both these energy components go into ATP synthesis. And this is readily testable by thinking about certain possibilities like prediction from this model and then testing it, and then we know that it works. Like for example, according to this model, the electron transport, or in other words oxidation, and phosphorylation both will be coupled. Okay, so that is the only way the proton is going to come down. So if we stop either one of them, like for example, let us say if you block this and the proton is not going to come. Now, as the electron transport is happening and as the proton concentration difference between the two sides keep increasing, you will reach an equilibrium. As long as the proton is not going to come, you can only build a certain concentration gradient where you will reach equilibrium between this concentration difference and the energy available from the electron flow. So as a result, the electron flow will end up stopping. So blocking phosphorylation will lead to oxidation, that is electron flow. That is one prediction and that has been observed. Okay. And so we see some experimental evidence here. For example, here, the oxidation or the electron flow is measured by measuring the amount of oxygen consumed. Okay. And also we measure the ATP produced for phosphorylation. So this red line follows the ATP concentration and the meaning phosphorylation activity, and the black follows the oxidation activity by measuring the oxygen consumption. So when you have the substrate ADP plus inorganic phosphate with no electron donor, you do not have anything happening to either one of the two processes. Then you provide a succinate. So remember complex 2. This can take transfer electron to quinone. So you can start the electron transport at this step. And now both oxidation, the electron flow, as well as phosphorylation both happen. And now when you stop the electron flow by adding cyanide, this is why cyanide is poisonous. So I didn't get into all the different inhibitors. They are all listed in tables in book and you can readily see them. They really don't need explanations. So when cyanide is added and cyanide blocks at this step, A to A3 in complex 4, and electron flow therefore stops, so oxygen consumption reduces. And the important point is when you stop that electron flow or the oxidation, automatically ATP synthesis also stops. Okay, so showing that these two are coupled. And this is what you would expect by this model. So you need the electron transport to build the proton gradient, and without proton gradient, you are not going to do phosphorylation. And that's what this experiment, so that prediction is consistent with this observation. And in the next case, what we are going to do is slightly complex experiment. So you have added the substrate electron donor, but nothing happens unless otherwise you allow phosphorylation possible by adding ADP and inorganic phosphate. And once you have added, so like in the previous experiment when both these two are available, that is the electron donor and the substrate for phosphorylation, now both processes start. And once you inhibit the electron transport, in this case instead of cyanide, in this experiment oligomycin or this venturidicin are added. So now the ATP synthesis stops and oxygen consumption, that is electron transport, also stops. Now there are inhibitors, they are called uncouplers, okay, like this DNP. When you add, what actually they do is they allow a leakage of the protons across this membrane, okay, and when that's like short circuiting this circuit. And when then that is allowed, now electron transport continues. Oxidation happens without any phosphorylation because you do not build a gradient, and without the gradient, there is nothing to flow through the turbine to produce a phosphorylation. Okay. So inhibitors of ATP synthase block electron transfer. However, there are certain agents which allow this uncoupling, then this happens. So this uncoupling does happen in nature. Like for example, in brown fat adipose tissue, this is a lipid deposition at the back of the neck in some of the organisms that live in very cold weather. There, the process is uncoupled so that the oxidation is used to generate heat energy without synthesizing ATP, so that the animal can be kept warm. So that uncoupling happens. So another important point that comes from this is this second point listed here. So the first point I sort of already explained. Why does electron flow stop when ATP synthesis is blocked? That is because the proton concentration increases to a level where now the energy available from the electron transfer is only able to maintain the gradient and no more than that. So it reaches an equilibrium with the electron transfer and proton pumping. But the other one is, if this concentration gradient only is required for ATP synthesis and not actually a substrate oxidation, then even without oxidation, if you have this electrochemical gradient, then ATP synthesis should happen. Okay, so that is written here. If the role of electron transfer is to create an electrochemical potential through proton pumping, then an artificial proton gradient, that is without using the electron transport process, if we make an artificial proton gradient through other means, that also should drive ATP synthesis, and that has been experimentally confirmed. So this is how we know chemiosmos. This is what is the best explanation for what happens in our mitochondria, as well as later today we will see happening in the chloroplasts in the photosynthesis. Okay, so this is the chemiosmotic theory of oxidative phosphorylation in our mitochondria. So now let us go and see what actually this is. What is this pump and how does this make ATP? So this has two subunits as you see here. One is FO. Okay, do not read it as F0 or F0, because this is not 0, it is O, and O stands for oligomycin sensitive. So this is sensitive to oligomycin. So oligomycin blocks this proton flow, and as a result ATP synthesis will not happen. So this subunit is present on the membrane. So it has hydrophobic residues that can interact with the hydrophobic lipid, and it is anchored on the membrane. So this is a cross section. Okay, so it is like a round structure and it is cut open to show the inner tubing or lumen. And then you have F1. So this is the catalytic subunit. This is where ATP synthase activity is located. So the proton flowing through this energizes this activity of F1. And details of these two are what we are going to see now. So this F1, the catalytic component, was first purified and shown to have the catalytic activity by Ephraim Racker, and he did this as early as 1960s. Okay, and an interesting experiment that he did led to a conundrum about the way the F1 ATP synthase works, and that was solved only in mid-90s, okay, 35 years later. That is because they needed to really crystallize and needed a leap in our understanding. You know, that is where you really call genius in thinking. It's not incremental process to propose a model to explain how the ATP synthase enzyme functions. So that will be our primary focus for today's class. So what Raker found was, when you incubate the purified F1 with ADP and inorganic phosphate, which is inorganic phosphate, and water with radio labeled oxygen, O18 water. Okay. So very quickly what he found was that all the four oxygens of the inorganic phosphate that would form if the original inorganic phosphate, non-radio labeled one, combines with ADP and forms ATP, and then that ATP hydrolyzes using the oxygen from the water, and this process happens multiple times, then only this phosphate will end up having O18 oxygen in all the four oxygen atoms here. And he saw that, and that indicated this explanation. That is, in the active side of this enzyme, this ADP plus PA forming ATP, and then the ATP hydrolyzing back to ADP and inorganic phosphate must be happening very quickly and multiple times. In other words, this process must be existing in equilibrium. Okay, so that is what he found. So its catalysis of ATP synthesis from ADP and PA is readily reversible. So this led to a problem. So then why would you need energy becomes an issue. Okay, so that is explained in this reaction coordinate graph. So for a normal enzyme, when enzyme substrate combines, this ES complex formation is the one that requires a lot of energy. That is where all the binding energy, non-covalent interaction, everything comes. Okay. On the other hand, with the ATP synthase, this particular ATP synthase, this mitochondrial F1 ATP synthase, that complex formation like ES, okay, so ES and then EP. So these are not having any big energy difference. It's nearly the equilibrium constant is 1. But on the other hand, for the ATP to dissociate away from the enzyme required a lot of energy. Okay, so that is an interesting difference from this. And why is this and what is the explanation for this took a lot of time. So what eventually people found was that the enzyme's active site very tightly bound ATP, and that itself was enough to drive the reaction from ADP plus inorganic phosphate to ATP. But the energy was actually required to release the ATP from the enzyme site, active site. And a mechanism for this, how this happens, was proposed by Paul D. Boyer, who won Nobel Prize for that in a really great discovery. So that we come to it in a minute after we understand the crystal structure of the F1 ATPase. And that was solved by John Walker, 1994. Okay, so the enzyme was purified 1960, and its crystal structure became available in 94. Because it's not that solving this crystal structure took 34 years. The crystallographic techniques became advanced enough to attempt these kind of complex structures. That process took that much time, and definitely this was one of the complex structures, and it required a lot of effort to solve the structure. So this is the model of the solved structure. So where you have the subunits here, so you have an alpha subunit and beta subunit, and they exist as dimers, and 3 such heterodimers are present. And then the gamma subunit forms a rod-shaped structure in the middle of the circular arrangement of these heterodimers. Okay, these are like 1, 2, 3. So you have 3 heterodimers arranged in a circular form, and in the middle, like a shaft, you have the gamma subunit. And this is the ball and stick model of this. And here, other subunits, there are a few more subunits, and they are not shown here. And FO is also not shown. And here you can see, to beta subunit, you have the ATP here, and then here to another beta subunit, you have the ADP bound. So these are shown in yellow and red colors. So this is a side view, like as shown in this model. And this one is as we are viewing from top down, okay, in this direction. So you have these arrangements, okay. And this, what is alpha MT, beta MT, all this will become clear when we go and consider the Boyer's mechanism. So pay attention in any case to these. These two have a conformation that is different from the other two, indicated by this outer line. Okay. So alpha, beta, but the conformation of the three heterodimers are distinct. Each one is different from the other two. Okay. So this is sort of indicated by in this cartoon by this round shape, and this is little sharpened, and this is with these sharp ends. So they have distinct conformation, and that becomes critical. So this is shown along with the FO subunit, which is like a barrel shaped C subunit. So there we are going to use the A, B, C. Like the C subunit, the 10 of them form the cylinder like structure on the membrane. And then you have the A subunit, and then the B is the one that contacts this F1 by attaching to the delta subunit, which is not shown in the structure of F1, the crystal structure of F1. And thereby it anchors this circular structure to the membrane. So this does not have any ability to rotate or move because this anchors tightly to the membrane. Okay. And what Paul Boyer proposed, let me show this picture, yeah, here. So he died I think 3 years ago, or I think 3 years ago, or maybe 2 years ago. Very recently he passed away. He is one of the pioneering biochemists. He edited Methods in Enzymology, several volumes of that book. So he proposed a model whereby as the protons flow through this cylinder, that is the FO's C subunits, that starts to rotate. And to that, via this epsilon subunit, the gamma subunit of the shaft of F1 is attached. And due to that, along with the cylinder, the shaft also rotates. And the shaft rotating, so this has specific contacts to the beta subunit. And as it rotates, it sequentially changes the conformation of the three beta subunits. Okay, so that is his proposal. This is called rotational catalysis. Okay, and that rotation cartooned here. So where you have this interacting with a particular beta subunit, this now undergoes a conformation change that releases the ATP out. And the interactions among them are such that when the gamma subunits interacts with a given beta subunit, changing its conformation to empty, meaning there is no ADP or ATP bound. That is why it is called beta empty, alpha empty. And when it does that, adjacent to one of them, conformation changes such that it binds ADP and inorganic phosphate. And since this readily becomes ATP from Ephraim Racker's experiment we know, then this changes conformation to this one. Okay. And when it again rotates, then as shown here, the next one becomes empty. The ATP gets released. And when it rotates to the next one, that also releases. So when this gamma subunit finishes one 360-degree rotation, each subunit have catalyzed the formation of an ATP and released it out as well. Okay. So the phosphorylation readily happens, and the energy required is only to drive the conformation to the empty conformation where the ATP is thrown away. Okay, and that is what happens. So that is shown in this animation, which I'll see whether I can play this. Okay, it doesn't play. So essentially what is going to see is this black arrow is the gamma subunit. So when it interacts with one given beta subunit, the ATP is released, and then now that is ready to bind ADP and PI. And that is what has happened here. And when it shifts to this, then this is going to become now the beta MT, and this will be the ADP binding one. And then ATP forms, and when it comes again, that is going to release. So this needle-like structure, you will see it rotating in jerky motion. Okay, every motion is 120 degrees. So the 360 is equally divided into 3 parts for the interaction from shifting from one beta to another beta to another beta. So it is not like a smooth rotation. Instead, it goes in steps. It's a 3-step rotation. And that is what happens. This is what Paul Boyer proposed, and this was confirmed later by really elegant experiments that this Yoshida and Kinoshita devised. So what these people actually devised is they made transgenic proteins alpha beta with the histidine residues, and the histidines, because of their negative charge, can bind to a glass slide. This is microscope slide coated with nickel. So this is anchored now to the glass slide, and you have the gamma subunit connecting to this cylinder C of this FO, and to that they have added biotin, this red circle here, and to that they add a long actin filament with avidin attached to it, and due to the avidin biotin affinity, this is now interacting with this. So now when you add ATP, and when this hydrolyzes it, so you have reverse rotation compared to the ATP synthesis, and that also would be predicted by Boyer's model, and that's exactly what happens. So now as it rotates, they had a fluorochrome attached to actin. So using a fluorescence microscope and time-lapse video, they were able to monitor the fluorescence shifting in a circular fashion, and they could see that it shifted in 120 degree at that time. And that is how they were able to observe how this happens. Okay. And here the energy available from the proton gradient and the energy required for the rotation is nearly perfectly matched, such that the machine, this motor, is at the theoretical maximum of energy efficiency in converting the proton gradient potential energy available in making this motion. And that is what made Boyer to call this as a splendid machine. So this is the amazing thing about an enzyme catalysis, where you have rotation of a turbine-like motion leading to the catalysis of ADP to ATP synthesis. So the main point is the energy required here is not for the reaction itself, but it is actually to release the product from the enzyme active site. So I hope this is clear. So with this, we are actually finishing our oxidative phosphorylation. So we saw electron transfer, that is oxidation, caused proton gradient, and also a charge separation. And when that potential energy difference is allowed to dissipate via a turbine, that is the F1 ATP synthase linked to this FO membrane bound actual turbine, we end up making the phosphorylation reaction, that is ADP plus inorganic phosphate becoming ATP. So this completes our understanding of starting from glucose, how we get energy in the form of ATP molecules, and we know how why ATP is high energy molecule, and we also know how the carbohydrate molecule has been oxidized to carbon dioxide, and in the same process oxygen being reduced to water. So carbon dioxide and water goes in the photosynthesis and carbon assimilation reaction that we are going to see next, and then finally it all comes back to carbon dioxide and water through the oxidative phosphorylation. So we have actually traveled with the electrons all the way from glucose to oxygen becoming water. So this completes one sequence of events, and through the process of this we have understood quite a few of the central concepts of biochemistry, including catalysis, bioenergetics, regulation, many aspects of enzyme mechanisms, and so on. So next we switch gears to considering a proton gradient formation where energy is actually input in the form of sun's light energy. Okay, here energy is released through the process because we have electrons falling from up to a down level, that is from lower reduction potential to higher reduction potential, and therefore energy is available. Now what we are going to do is we are going to provide energy and force electrons to flow through to make the proton gradient again. Okay, so that is what is photosynthesis. So before we get into those details, let us have a global picture of matter cycling among the biological systems, that is between photosynthetic organisms and heterotrophs like us. So that part we'll look at in this slide. So this is extremely simple. You probably learned in high school, but probably did not look at it from a philosophical angle. So that is what I want you to do here. So the sun's energy drives the photosynthetic cells to combine carbon dioxide and water to produce carbohydrates and evolve oxygen in the process. Okay. So carbohydrate, you have the three elements required. It's amazingly simple. Only three elements that we put together to make life happen. So the energy production, liberation, and most of the storage all are involving only these three elements. Extremely simple system. So photosynthetic cells put these together to make this, and we just saw the heterotrophs consume these and return these back. So therefore in living system, by the action of sun's energy, this is the sole source of energy. Okay, if you are burning fossil fuel is carbohydrate made millions of years ago or thousands of years ago, and that was made by photosynthetic cells. So sun's energy is the only source of energy in the biosphere, in the biological systems for the carbon, hydrogen, oxygen cycling that is shown here. This cycling is made possible by the energy from sun and sun alone. So the equation above summarizes the process. So the energy of light, CO2 plus H2O gives you CH2O plus O2. So now let's get into some of the details in the next few minutes of time we have. So this slide summarizes the general features, like when you have light, through light reactions, because it's light dependent, so we call them light reactions. Water is split and electrons are taken up, so oxygen is returned. So the water is here oxidized. Just now in the previous one we saw the opposite. So here, this is one process. Second, you have these oxidized coenzymes like NAD. Photosystem uses the phosphorylated version of NAD, so NADP. Otherwise, here also the electron transfer is in the hydride form, gets reduced to NADPH. So oxygen is evolved and reducing equivalents are produced. And the third is ATP formation as well. Okay, so these are the things the light reaction produce. And carbon assimilation reactions, do not call them dark reactions because they can happen even in the presence of light. It is just that these reactions are not dependent on light. So therefore, the correct way of calling them is carbon assimilation reaction because it assimilates carbon dioxide. So by using the reducing equivalents and the energy in ATP, these reactions reduce carbon dioxide into carbohydrates. So therefore, you have two components, one light reaction producing ATP and the reducing equivalents. Okay, oxygen evolution is incidental in terms of our energy here. And these two are used to convert carbon dioxide to carbohydrate. So essentially, whatever is the end product of TCA cycle, decarboxylations we saw producing carbon dioxide. And here now we take the carbon dioxide and make carbohydrate. So these are the two major components of the photophosphorylation reaction. So 2 points I want to draw your attention to. One is in the mitochondrial oxidative phosphorylation, the electron donor is NADH. Okay, so that is a very willing donor of electrons compared to the molecule that is going to donate here, which is water. So therefore, to get electrons out of water, you really need energy. So that is one important difference here. And that energy is what is coming from sun. So how is that energy used, okay, abstracted to do this work is one important thing we are going to see. And the second is that you need which I just mentioned as first part of it itself. The first part therefore is just that NADH is a good electron donor and water is not. So and water is the electron donor here in the light reaction. And the second, as a result of that, you need energy input. So photophosphorylation differs in that respect from the oxidative phosphorylation. Oxidative phosphorylation did not require energy input. We did not hydrolyze any ATP to move the electrons from one complex to another complex. Whereas here energy will be required, okay, to generate good electron donor as well as good electron acceptor. To do that, you need energy input. So these are the two main differences we need to keep in mind. So I'll stop here with these, bird's eye view of photophosphorylation, and we'll get into the details tomorrow. So the first thing is we are going to look at how is light energy taken up to really generate a good electron donor and a good electron acceptor if possible, that we may not be able to finish tomorrow. So that is where we are going to focus tomorrow.